Begin, look at what enzymes are, and so these are classifications of proteins. There is one exception of ribozymes that are made strictly of RNA, but they serve as biological catalysts, and what they do is that they increase the rate of a reaction. They are not changed by the reaction, so they can be used again and again. So what this means is that the enzyme itself is not changed at the end of the reaction. It just helps to increase the rate of a reaction within the body. They do not change the nature of the reaction. The reaction will occur without the enzyme. It just will occur at a slower rate without the enzyme. And then also that means that that does not change the nature of the reaction or the final result. And so the result will always be the result, however, the speed of which we get there just increases. And so enzymes too, what they do is they lower the activation energy for the reaction to occur, thus they occur more rapidly. The activation energy is the energy that is required for the reactants to engage in a reaction. So again, enzymes are what decrease this activation energy. And so most molecules lack activation energy for a reaction. But by adding heat, this will increase the likelihood of a reaction to occur, and so will increase the rate of the reaction. But there is a limit as to how hot something or how much heat something can have without it being detrimental. And so catalysts help the reaction when it gets to be too hot and helps to lower the temperature. If we look at this picture here, let's first start with a non-catalyzed reaction. All right, so this is showing, let's look at the bottom panel here, that this red wall is the activation energy, and so the reactants have to have a large amount of activation energy, so increase the amount of energy they have to get to their final product. However, when we have a catalyzed reaction, this decreases that wall that we have for that activation energy, and so it's easier for those reactants to formulate their products, okay? So if we look at the top panel, so again, here's the non-catalyzed reaction. So energy of reactants requires a lot of energy in order for that reaction to occur, whereas in a catalyzed reaction, then we need less energy for that reaction to occur to make our final product. So, like with everything for anatomy and physiology, the function of the enzyme is dictated by the structure. And so each enzyme has a characteristic 3D shape or conformation with pockets that serve as active sites in the enzyme. And so this is what we term as a lock and key model. The reactants are called the substrates. Okay, so here are our reactants A and B. And they fit into active sites like a lock and key model. So substrate A, say, fits in this one, and then substrate B, or reactant B, will fit in that spot. And so sometimes the initial fit is not exact, but will change as the substrate moves to that active site. Again, because these are not static, and so these might conform to help make the correct shape. And so when the substrate binds to the active site of an enzyme, it forms a temporary bond, and it's weakening the original bond of the substrate, allowing them to break easily. So then new bonds can be formed, and they're brought closer together by the enzyme. And so bonding of the enzyme, again, makes a temporary substrate enzyme complex, and then this breaks to yield the product. All right, so now here we have our new product of C and D in this example. And so the amount of the enzyme in a sample of fluid can be measured based on the rate of the product synthesis. And so it tells us how effective are these enzymes that we have within our body. Another example that we have is called an induced fit. Okay, and so with this, what we have is that the enzyme and the substrate, so here's our substrate, this would be our enzyme, is going to form during the course of the reaction and then dissociate to yield products and then free the unaltered enzyme. So even though it doesn't look like it will fit in this picture, what changes is this active site to make that fit, where we're inducing a fit for that substrate. And so this occurs with the enzyme lysozyme, and it helps to kill bacteria. So the fit here, like you can see, isn't perfect at first, but then the enzyme will change shape to fit that polysaccharide substrate in, and so that it can increase the rate of that reaction. Typically, we see enzymes having the suffix of ase, and so phosphatases are examples, and so these will remove phosphate groups. Synthases and synthetases catalyze dehydration synthesis. Hydrolases promote hydrolysis. Dehydrogenases remove hydrogen. Kinases add phosphate groups, and these are very important, especially for our nervous system. And then isomerases rearrange atoms into different isozymes. Isoenzymes are enzymes that have different amino acid sequences. So these enzymes are named by their activity regardless of where they are in the body. It does not matter. So examples of diagnostic measures of doing testing for enzymes within the blood. So for example, I'm sure some of you have heard of amylase, and so this is to test for pancreatitis and then peptic ulcers, which is important for breakdown of carbohydrates. Creatinine kinase or creatinine phosphokinase, CPK. This is testing for problems within the muscle, so muscular dystrophy and myocardial infarction. Also, lactate dehydrogenase is very common, where we have an increase in lactic acid, and so that would also signify damage within the heart, liver disease, renal disease, and possibly anemia as well. So the activity of an enzyme from going from substrate to product can be influenced by several different factors. So this one is looking at temperature in this graph. So here we have our enzyme activity on the y-axis, and then on the x-axis we have our temperature. So here within cold conditions we see very low activity within enzyme. But as we have an increase in temperature, then we also have an increase in enzyme activity. Okay, but there is a certain threshold in which we will start seeing a decrease in activity when it gets to be too hot. And so this is because, again, if you remember, most enzymes are proteins, and so this will cause them to be denatured, thus being ineffective. The next influence for enzyme activity has to do with pH. And so this graph, again, on the y-axis, we have our enzyme activity, and here we have pH. And so enzymes are found within all over the body, and it doesn't matter what organ it's in. However, having a certain pH range will either activate or deactivate them. So for instance, with this one, pepsin, this is active when we have a low pH. And so this enzyme is found within the stomach where we have very acidic conditions, and so helps to break down nutrients. Now, this becomes inactive when we have a high pH, so say around 6, when we look at that green line, and it starts to really decrease. Salivary amylase, so its peak is around 7, and so we find this within the mouth, so very neutral. But then when it's too acidic or too basic, then we'll have it not working anymore. And then lastly, what we have is trypsin, and so its peak is around 9.5, and 2, if it's too acidic or too basic, then we start seeing a decrease in the activity of that enzyme. And so trypsin, we find this within the small intestine, helps with the breakdown of proteins as well. So coenzymes are also important for enzyme activity and for increasing them, because most enzymes need additional small molecules to help them in aiding with their reaction. And so coenzymes are going to be some type of organic molecule that will be derived from vitamin soluble or water soluble vitamins. And so what they do then is transport hydrogen atoms and other small molecules between enzymes. So in this example, here would be our coenzyme, and this is our actual enzyme. And so that when they bind together, then this enzyme can have its active site open and ready for the substrate. And so if we look at this graph here, this blue line represents only the enzyme, and this pinkish line represents the coenzyme with the enzyme. And so you can see the rate of the reaction is higher than with the coenzyme than it is by the enzyme itself. Cofactors are typically metal ions such as calcium, magnesium, manganese, copper, and zinc that also help to form the active site through a conformational change of the enzyme or helps in enzyme substrate binding. So here in this example, what we have is zinc is going to bind with that enzyme, and so that's going to allow then for the substrates to form and bind at the active site to yield then their different products. Another factor to consider is the actual amount of substrate that is present will also affect the reaction or the enzyme reaction. And so what we have here is, again, on the y-axis reaction rate, but now we have the substrate concentration. And so as the rate of the reaction increases until the enzyme becomes saturated. And so this is termed as a maximum rate and also as saturation. So once we hit saturation, that means that there are not enough enzymes present to increase that speed of that reaction. So no matter how much more substrate you add, the product result will still be the same in terms of how fast that will occur. And so at this point, you will not see any more reactions occurring at a faster rate. Now our enzymes do not hang around forever because, again, they are proteins, and so they are subject to being degraded. And this is due to breakdown and resynthesis of enzymes, and a lot of it does take energy, and so we're continuously creating and making new enzymes to replace old ones. So this allows genes to alter enzyme activity and metabolism of our cells, and so we especially see this with aging, where we have our slower metabolism, and that's really due to a lot of enzymes that are starting to break down and not be formed as quickly as they were when we were younger. Sometimes a single enzyme can drive a reaction in two directions, and so this depends on the concentration of substrate or the product, interchangeable. And so this is what's termed as the law of mass action, where it has to do with reversible reactions. So when one side gets higher, the other reaction reverses. So if we have, say, an increase in carbon dioxide, then we'll also have an increase in carbonic acid. And so for this example, that's going to be increased by the enzyme carbonic anhydrase. However, at some point, there will be saturation of the substrate and will not affect the rate of the reaction. Most reactions are linked together in a chain or web called a metabolic pathway. And so these begin with an initial substrate. So here in this example, our initial substrate is both with A, and end with some type of final product. Okay, and so there can be several or many enzymatic steps that occur along the way. So this top example is very actually uncommon, even though it's the easiest to understand, where here we have the substrate is going to have one enzyme create this B intermediate, a second enzyme creates another intermediate, third enzyme creates another intermediate, and so on until we get our final product. In reality, most of our reactions occur in this type of fashion. So we have our initial substrate where we'll have the activation of the first enzyme, creates one intermediate. Okay, then it will activate another enzyme. However, then this third intermediate will be activated by this type of enzyme and then this type of enzyme, or then we'll have two different intermediates continuing on, and so ultimately we'll have two different final products. And sometimes these are biologically necessary, but sometimes these can yield overexpression of products, and so could be problematic. So could be some type of disease or some type of cancer where it could have a negative effect for us. With those metabolic pathways, what we also get into are end product inhibition. And so here's a classic example of homeostasis as well, so that we don't have too much of something. And so what we have here are branch points are often inhibited by a form of negative feedback in which one of the final products inhibits the branch point enzyme. So once we have enough of a concentration of this second final product, then what that will trigger then is for, say, this enzyme to stop working. And so thus, when we have the intermediate of, say, C in this example, then we'll continue with the production of, say, this final product, okay? And so this whole process then is called allosteric inhibition, where the product binds to the enzyme at a location away from the active site and changes the 3D conformation of that enzyme. And so this keeps the final product from accumulating. And so let's put it in layman's terms. So for example, in a factory, if too much of the product won't affect the output of the workers and the products. Okay, so no matter how much of that product that you have, there's only a limited amount as to how much can actually be produced by those workers. And so this is similar in terms of what is going on within the body. Our genetics can also affect metabolism and the activation of and expression of different enzymes. And so this is now we're going into inborn errors of metabolism, where a mutation in maybe just one gene that codes for an enzyme in a metabolic pathway can be affected. And so the products to be formed after this enzyme of the chain are not formed. Okay, so here in this example, what we have is enzyme number 3 here. This has, say, one mutation, and so does not produce this final product anymore. Rather, it creates the second byproduct, and so again, this could cause then different types of diseases. Due to an accumulation of, say, maybe some of these intermediate products, or it can be an alternative branch that we see. And so, for instance, PKU is an example of an inborn error of metabolism. So let's go to the next slide, where here are more examples where like albinism is an example where we have lack of melanin. Lactose intolerance, this has to do with enzyme that produces a faulty product. Tay-Sachs disease, this occurs within the nervous system, and we have lipid accumulation, and so children actually don't survive past the age of 5. Also hypercholesteremia, high blood cholesterol levels leading to atherosclerosis. So these are examples of where one enzyme can have just one genetic mutation, causing then some significant disease later on in life.